Hi students, how are you? I hope each one among you are doing great and well. Welcome to Supnit's classes. This is Rahmat Latif, your biology educator. In this session, in this video, I am going to talk about some outbreeding devices through which inbreeding will get, overcome. Inbreeding depression simply means when sexual intercourse or when reproduction, fertilization took place between two closely related, partners, or we can say two closely related flowers. Say, for example, we are having same flower in which male and female reproductive structure is located, and that flower is known as, monoecious flower, bisexual flower. Okay, what will happen in that? Autogamy happens. Pollen from same flower comes in contact with the stigma of the same flower. And what happens during that? Inbreeding occurs. Inbreeding simply means that depression, inbreeding depression is that depression due to which diseases often occur to happen. Okay, like those diseases which are, recessive, they become dominant over some species, over some generation, sorry, over some generations. In order to avoid that inbreeding, we just use outbreeding devices. And those devices are those, they are not only they are not external devices. What we do, we just try to prevent inbreeding through outbreeding devices. We just try to prevent inbreeding. We just try to prevent inbreeding. Okay, what we will do, we in some species, what happen, pollen release and stigma receptivity are not synchronized. What I actually mean to say, for example, this is flower and it contains both female reproductive structure and male reproductive structure. Okay, and in this female and male reproductive structure, synchronization doesn't occur. It means when pollen becomes mature, stigma, female reproductive structure is immature, or vice versa, and autogamy will be reduced, right? Autogamy won't happen in such case. Since autogamy won't happen, what will happen then? Neither inbreeding depression will occur, right? Neither inbreeding depression will occur. So it's, beneficial for us that is autogamy doesn't happen and inbreeding depression will not occur. We will try to make all the flowers like that, that is, they should not get, synchronized, they should not be synchronized, that stigma receptivity and pollen release should not be synchronized, so that our flower will avoid inbreeding depression. Okay, in some species, what will happen, what happens in some flowers, position is different. Say, for example, female reproductive structure, that is, part of female reproductive structure, stigma is here and anther is here, so it can't reach here, right? Or say, for example, it's here, female reproductive structure is here and male reproductive structure is here. That means positions are different. Since positions are different, again inbreeding depression will be, avoided, because it won't reproduce with the same, with the stigma of the same flower. Okay, these were the two methods in which pollen synchronization doesn't happen, and another method was in which basically, you know what happened, basically positions were different, right? Now we will see another one is genetic mechanism. In genetic mechanism, what happens basically? Mechanism, say, for example, here pollens are released. Okay, but what will this stigma do? It won't allow growth of pollen tube. It won't allow growth of pollen tube. Since pollen tube will not grow, that means male gametes will not be passed to the ovary. This is the genetic mechanism. Okay, I hope this concept is clear, right? These were the three methods. Now we will see how fertilization occurs. Say, for example, this is our female reproductive structure. I have to explain here pollen pistil interaction. Pollen pistil interaction, okay? Say for example, pollens are released here, okay? Pollens are released here. What will these pollens do? They will simply, after release of pollen on the stigma, after getting stickled to the stigma, pollen tube growth will take place, which will take it to the, you know, egg, which is inside the ovary, okay? Pollen tube growth will occur. And after this pollen tube growth, it will carry male gametes. As I already mentioned, 60% what happens in 60% angiosperms, what happens in 40% angiosperms. So here we have two male gametes, right? Because one pollen grain carries two male gametes. So two male gametes will get through this pollen tube. Now the question is, what is the pollen pistil interaction? As the name said, it's the interaction between pistil and pollen in order to release male gametes near egg. What happens basically, female reproductive structure, particularly synergids. I'll tell you what synergids are. As I already mentioned, synergids are located at micropylar end. And what happens to those synergids? They are, located with egg, and egg cell and synergids together called as egg cell. Okay, and what happens? Those synergids release chemicals basically. Those synergids release chemicals to pollen, which allow pollen to, which allow this to grow pollen tube, and through this pollen tube male gametes are carried to the egg cell, which is located here. These two male gametes will reach here. This is pollen pistil interaction, okay? It's not rocket science. Pollen pistil interaction is the chemical interaction between pollen and pistil in order to grow pollen tube, through which male gametes will be transported to the egg cell through pollen grains, with the help of pollen grains. I hope this concept of pollen pistil interaction is clear. And as we know that pistil will recognize obviously pollen of its own species, not of another species, because we will learn this in cell. Cell has some, receptors. Membrane has some receptors, which actually, recognizes the type of, which actually recognizes the type of male gamete, or we can say pollen grain, whether it is of my species or of another species. Had it been of another species, it won't allow its growth. Had it been of its own species, it will allow its growth. I hope this concept of pollen pistil interaction is clear. What will happen then? See, hold on. What happens in flowering plants? In flowering plants, we have double fertilization. Okay, please remember. We have double fertilization. And double fertilization involves simple fertilization, or syngamy, plus triple fusion. I'll explain what is mm, fertilization and what is triple fusion. Okay, see, say for example, this is female reproductive structure, right? Okay, I won't make this whole, I will make only inside the structure which is inside megaspore, like this. Okay, here we have three cells, 1, 2 and 3. Here we have 1 and 2 and another cell. Here we have two nucleus. I'll make nucleuses by, we have 1, 2, 3, 4, 5 in a central cell, this like this. And here synergids contain filiform apparatus. I have explained this. Whosoever hasn't attended that video of mine, please go through that video and check that video and come back to this video. Okay, See what will happen. Pollen tube growth. Pollen tube, say for example, pollen tube comes like this is the structure inside the ovary. Please don't get confused. So pollen tube will get inside the ovary. It will contain two male gametes, right? It will contain two male gametes. Okay. It contains two male gametes. It's actually the interaction, the chemical interaction between this synergid and pollen. Synergid will release some chemicals which will show them the path where you have to go. Okay. Since the first male gamete which will be released here, or which will reach here firstly initially, that will break the synergid, okay, and finally comes in contact with, I'll make male gamete through blue, green one, okay, comes in contact with this egg cell. This is egg cell, okay, egg. It will come in contact with egg cell. Okay, since it comes with egg cell, it will get fused. Fusion will took place here. The fusion is known as fertilization, right? It's known as fertilization, and fertilization will give rise to zygote. Okay, since zygote will be formed here, we have one more male gamete, right? We have two male gametes. One will fuse with egg, which is known as fertilization. Another one will vent here with polar nuclei. One, two, three. Since it involves fusion of three nucleuses, this is nucleus of male gamete. These are the nucleuses, polar nuclei, which are located in central cell, as I already have mentioned, right? Since here we have three nucleuses, it is known as triple fusion. It is known as triple fusion. Okay, I hope this concept of triple fusion and fertilization is clear. Zygote will then get converted into embryo. This structure will get converted into endosperm. Okay, this will get converted into endosperm. This will get converted into embryo. Okay, I hope this concept is clear. Further, this endosperm will get converted into endospermic tissue, cellular endospermic tissue, or free nuclear endospermic tissue, free nuclear endosperm, which we will deal with upcoming session. Okay, since this involves two types of fertilization. One is simple fertilization, which occurs with egg cell, fusion of male gamete with egg cell, and another is fusion of nucleus with polar nuclei, which is located in the central cell. That is known as triple fusion. And triple fusion and fertilization will occur, okay, and that kind of fertilization is known as double fertilization. So in plants, double fertilization occurs. Please remember this concept. If anybody will ask you what kind of fertilization will occur in sexually reproduction flowering plants, that is double fertilization. Okay, I hope this concept is clear, right? Okay, we will see artificial hybridization now. Artificial hybridization is that hybridization which is done by man in order to avoid any kind of autogamy or heterogamy, in order to get variable seeds, in order to get genetically variant. Okay, in order to get genetically variants, right? We will see what is artificial hybridization, what we do in artificial hybridization. Say for example, this is our flower. This is our flower, okay, which contains both female reproductive structure and male reproductive structure, right? What will happen in this? Chances of autogamy is higher. Autogamy could happen, right? Autogamy, chance of autogamy is higher. So what we will do in order to avoid that autogamy, we will just cut its anther. Cut anther. But in which stage? Not in matured stage. In you know in young stage, okay, not in adult stage, in bud stage. Okay, we will cut this, and that cutting off anther is known as what? Emasculation. Okay, I'll write it down. This is known as emasculation. First step of artificial hybridization is emasculation. And what is emasculation? Emasculation simply means when we cut anther in its bud stage. Okay, why we cut that? In order to avoid autogamy, which can lead to what? Which can lead to inbreeding depression. Okay, in order to avoid that inbreeding depression, what we do, we just do emasculation. We will just cut anther, but not in its flower stage. We will cut anther in its bud stage. Okay, after cutting it, what we will do, we will just do bagging. Okay, we will second after emasculation, what we will do, we will do bagging. We will cover this flower with the help of butter paper. We will cover the flower with the help of butter paper. Okay, I hope this concept is clear. We will cover the flower with the help of butter paper. Okay, done. We just cover the flower with butter paper, and when the female reproductive structure, that is pistil, when that attains maturity, we will just remove this bag, we will just remove this butter paper, and we will sprinkle the desired pollen grains on the stigma in order to do artificial hybridization, in order to do that hybridization which will really benefit us. Okay, we will sprinkle the pollen grains on the stigma in order to do artificial hybridization, and after that we will again do rebagging, so that the desired pollens will only get, so that only desired pollens will come in contact with stigma of the female reproductive structure, or we can say pistil, and the pollen tube will grow and finally will reach to the egg. Okay, I hope this concept of bagging is clear. And after bagging, we also do rebagging. Okay, this is artificial hybridization. Why artificial hybrid? Why do we perform artificial hybridization? We perform artificial hybridization so that our we will get desired kind of seeds, we will get desired kind of flowers, we will get desired kind of hybrids, so that we will get benefited. Okay, I hope this concept of artificial, what we call it as hybridization, is clear, right? Great. Okay, we are done with artificial hybridization. We are done with what we call it as double fertilization. We are done with double fertilization. Great. We are done with pollen pistil interaction. We are done with outbreeding devices. I hope this concept of today's session is clear. I'll just make a recap of all this. Outbreeding devices simply are those devices which inhibits inbreeding depression, so that we will get desired, we will get desired seeds, flowers, or fruits. Okay, great. This was the work of outbreeding devices. Then we have pollen pistil interactions, the chemical interaction between pollen and pistil, so that pollen will release male gametes in the pollen tube, and the male gametes will exactly reach to the egg. Okay, exactly reach to its destiny. Okay, and pollen pistil interaction occurs when we don't have pollen. When pollen of same species doesn't come in contact with stigma, it will not allow its growth inside the pollen tube. And when pistil will found it's the pollen is of my same species, it will allow its growth through pollen tube. Okay, great. Okay, done. We saw double fertilization. I'll again explain this what double fertilization is. Since one pollen grain contains two male gametes, one male gamete will come in contact with the egg and it will form zygote here, which will further get converted into embryo, and another male gamete will directly go to the polar nuclei and form three nucleuses there. That's why first fertilization is known as simply fertilization and another one is known as triple fusion. It involves three nucleuses, one, two, and 3. Since two types of fertilization occurs, one is simple fertilization and another one is triple fusion, so in total we can call it as double fertilization. Okay, I hope this concept of double fertilization is clear. And then we have artificial hybridization. What we see in artificial hybridization, simply what happens, you know. We just hybridize artificially in order to get more desired seeds, more desired flowers or fruits or plants. Okay, what we do, we just first of all we do emasculation. Emasculation simply means when we cut anther in its bud stage in order to avoid autogamy, which can lead to in inbreeding depression. Okay, after emasculation we cut anther, and the question is if there is not the flower which contains both male and female reproductive structure, then we don't have to perform emasculation, obviously, because we can't cut any anther there, because flower is unisexual, flower is dioecious, that's why we can't perform emasculation. After this we have to perform bagging. We have to cover the flower with the help of butter paper, so that we have to cover the flower with butter paper. What's the function of butter paper? It will protect our flower, it will protect our female reproductive structure in order to get in order to avoid fertilization, or we can say pollination with undesired pollens. After, when our female reproductive structure will attain maturity, what will we do? We will just simply put the pollens, you know we will just put the pollens, we will put the desired pollens on the pollen tube, sorry, stigma, and we will again do rebagging. We will just cover that with butter paper in order to avoid further, in order to avoid that undesired pollen should not come in contact with the stigma, so that our female or our egg will get fertilized by our desired pollens, which we sprinkle on the stigma. I hope this concept is clear. I'll meet you in such any other session. Till then, goodbye. Take care. Thank you. Thank you so much.